Hello, listeners. Yamina here. Welcome to the Dr. GPCR podcast. Before we dive into this episode, I have a few announcements to make. First, I'd like to thank you all for listening in every week to these podcast episodes. It's been 4 years since we started recording podcast episodes. Actually, February 18th marks the fourth anniversary of recording our first episode with Dr. Paul Insel. This month, we also launched the Dr. GPCR University program to provide courses and content on all aspects of GPCR research to support our community. To kick off this new initiative, we partnered with Dr. Terry Kinakin, who teaches the course Applying Pharmacology to Drug Discovery. Due to an overwhelming response and demand for this course, we have decided to bring it back later on this year, so stay tuned. To get more information about the Dr. GPCR University and what we do, please join the ecosystem for free today at drgpcr.com. What are you doing on April 22nd and 23rd this year? Join me and our GPCR colleagues for the inaugural Endocrine Metabolic GPCRs meeting taking place in Birmingham in the UK. This event will bring together scientists from both academia and industry to exchange data, technologies, and ideas in the field of GPCRs. Some key topics include the latest GPCR signaling methodologies, metabolite sensing GPCR biology, novel endocrine mediators, and GPCR activation structures. This meeting is organized by Bioscientifica and endorsed by the Society of Endocrinology. We are very grateful for Bioscientifica for having the opportunity to partner with them. I will be there in person, and we will also have a Dr. GPCR booth. So stop by the booth, say hi, and hopefully you'll enjoy the meeting as well. Mark your calendars for the next season of the Dr. GPCR Symposia. And more specifically, mark your calendar for our first event of this year on March 15th. This first symposium will cover the topic of GPCR activation and signaling. We will have talks. We will also be able to accommodate posters on Zoom using the breakout rooms. You can visit the Symposia page, which is already up with the latest speaker information to stay up to date and get to know our speakers. If you'd like to give a talk or present a poster, please email us at hello@drgpcr.com. We will not be doing poster selection. Everybody is welcome to present their poster. We have 99 breakout rooms, so we can accommodate 99 posters. And now let's dive into this episode. Hello, everyone. This is Yamina from Dr. GPCR. It's very early morning here in the Boston area, and I'm very happy to have with me Dr. Tobi Langenhan. Tobi, welcome to the podcast.
Thank you very much. Hello.
Very excited to have you on. So let's start at the beginning. Who are you and what do you do?
Yeah, so I wear different hats actually professionally. I'm a scientist from Germany. I'm a medical doctor. I'm a biochemist, a professor of biochemistry, but I'm also a neurobiologist based on the fact that the research questions that have to do with the GPCRs we're going to talk about are related to the undertakings and the functions of the nervous system.
Wow, so many hats. So let's deconvolute and let's start at the beginning. Did you always know you wanted to be a scientist?
Yeah, kind of. So I enrolled in medical school with, I think, the idea already embedded in my head that whatever I learn, I will actually put forward to a career, or at least the idea that I then had as a kid or as a teenager of a scientist. If I look back, I accomplished at least this. And so when I stopped, or when I graduated from medical school, I already had undertaken research projects, which is customary in German medical schools during the curriculum. And I was researching neuroanatomy, and in particular the olfactory system of anatomy. That was around the time when the Nobel Prize was awarded to the discovery of the large GPCR class that encode the olfactory receptors. And that somehow convinced me finally actually to embark on further stations in science, in research, rather than joining my colleagues at the clinic.
So you completed medical school and then switched completely to research.
Right. I switched completely to research. So I had one conviction. I wanted to actually get better educated in neurobiology because this was the area that I did my MD degree in. So I applied for a couple of scholarships in the US and the UK, and accepted one that came with a dual course in as a master's and a PhD, or a DPhil, how it's called, at Oxford University. So I went from Germany then to Oxford for 5 years and studied neuroscience, and also did a DPhil degree, a PhD degree in neuroscience with particular focus on biochemistry and in particular the biochemistry of adhesion G protein coupled receptors. That's how I ended up with GPCRs in the first place too.
Did you from your interest in neuroscience, there is a little bit of distance between neuroscience and GPCRs. You cannot go walk through, look at a neuron and not bump into a GPCR. Was the interest more on the neuroscience side, and then you discover GPCRs, or the other way around?
No. So I was mainly 100% interested in neuroscience, and by serendipity, actually, I got handed over a project that became my doctoral project that introduced me or connected me to G protein coupled receptors. So I was doing my project at the Department of Biochemistry at Oxford in a lab that was led by another German doctor, Andreas Russ, and he just set up shop there. And he hailed from the University of Cambridge, the lab of Martin Evans. And so they were one of the top labs back then in the late 90s, early 2000s, that could genetically target ES cells and generate basically transgenic mice. And he identified G protein coupled receptors as one of the big druggable genes in the genome, and basically founded a company to generate mouse models, genetic mouse models, to study the expression and also the phenotypes, the null phenotypes of G protein coupled receptors. And then after a while in industry, decided to actually establish an academic lab at Oxford and go for the very few mouse models that weren't basically purchased by industry. And it happened to be adhesion GPCRs because they were just much more too complicated to be worked at mainly by the big pharma companies back then. And so that's what he ended up with. And then he also knew that back then, 20 years ago, mouse genetics is a very laborious, very expensive, and very long endeavor. So he actually established on the side in his lab an own a different basically regime of experiments dealing with C. elegans. So he analyzed orthologs from adhesion GPCRs in mice or in humans, also in the roundworm, in a nematode, Caenorhabditis elegans. And I was his first PhD student, and he actually handed me over a strain of worms, of which he only knew contained a mutation in one of these genes, and it happened to be an adhesion GPCR. And that's actually how I was connected and picked up my very first GPCR project. So complete serendipity. Could have been anything else.
Wow, I love it. I love it. You mentioned applying for different scholarships and then ending up at Oxford for your DPhil. How did you pick the lab, or what were the criteria? And before you answer, the reason I'm asking this is because a lot of our people who listen to the podcast are between the ages of 25 and 35. So I think learning more about how others have made their decision to join a particular lab could be very useful.
Yeah, so the way this master's DPhil program was structured in the UK, in Oxford, was very special back at the time, and I think very recommendable, because within this years of masters, we were sent to different labs to do our master's thesis. So I did actually 2 master's theses by research in 2 different labs, which gave you a very intricate look into the science that was done at a particular lab first, but second also basically the atmosphere, how You could cope with the people, with the lab head, with the postdocs, with the fellow students, graduate students there. And that's actually when I decided actually that I'm going to join this particular lab, because I liked the atmosphere. I liked the fact that this lab picked up a very say unfashionable part of research, and it was unfashionable, by all means. So much of the research we did in the first years was not very popular, because it was very rare that somebody actually looked into adhesion GPCRs. It was a lot of new concepts. We were trying to figure out our way, and that appealed to me. So I didn't want to join a big and established lab where I would be one of the students that worked on a project carved out 2 years in advance. I wanted to do something that was pretty new, also at the risk, of course, of that might fail, but because I'm a medical doctor, I could, I was a risk taker, because I wasn't afraid of failing in that particular research project, because at any time I could have actually returned to a clinical routine and become a doctor.
I love it. High risk and high reward.
Yeah,
fantastic.
sort of.
So
Yeah.
you get your PhD, you get introduced to adhesion GPCRs, working in C. elegans. What then?
Well, then two things happened. So I returned to Germany because I was already married back at the time, and my wife couldn't come over to the UK. So, I moved back after 5 years of long distance relationship, and set up my first lab at the University of Würzburg, at the Department of Neurophysiology. And I picked this particular lab because I got hooked on adhesion GPCRs, and I wanted to collaborate with somebody who could teach me electrophysiology, because I was interested in adhesion GPCRs in the central nervous system, or in the nervous system, in an invertebrate model, because of all the advantages that invertebrate models actually bear. And so I switched model organisms from C. elegans to Drosophila melanogaster because of the fact that you can do proper electrophysiological analyses at a much broader and deeper degree in Drosophila as opposed to C. elegans. And so I joined this lab there, started my group, wrote the first grant, and luckily got it accepted. So started off with my first PhD student maybe a year into that period, basically. That was the first thing. And the second thing that happened, which I think was very important for my own career, is that my doctoral advisor in Oxford decided to actually finish his academic career. So he closed his lab after, I don't know, 10 years in business, moved back to Germany and completed his residency. And ever since, he is a general practitioner in a rural area in Germany. And that's very important because I guess it was very important for me, because that meant that I didn't have to basically graduate from the research projects that I did in my PhD. There wasn't any competition I would have to have avoided, because we just left. So I took over a couple of the projects and continued ever since, which admittedly is quite an unusual an unusual way actually to go through the different levels of academia. Yeah.
Well, and how interesting is that? I mean, it's also, I love the idea of taking over some of these projects. I think it was a good move. But how interesting is it to end your career as an academic, finish, and then become, well, it's a good plan if it fits your needs.
Absolutely.
But it's
So
absolutely unusual.
it's admirable, it's fearless, and it's successful. So I think it deserves to that the word is spread about it. I mean, this specifically in our times now, I don't think it's very wise actually to write down your career from your 20s till your mid-60s or in your 70s, because a lot of things can happen. You should be prepared, or at least open to crossroads, and maybe take the other road.
I agree, and I think I was expecting you to mention that he actually went into industry,
No,
not back into finishing. Wow. Well, it's great. I
no,
think it's a
yeah,
good sign. I think the take-home message for the audience is follow your passion, follow whatever you feel like you want to do. And
yeah,
if
and
one thing doesn't pan out or you don't feel like happy in your current space, just find where you're happy.
I agree, absolutely, yeah.
Sorry, I didn't mean to interrupt you.
Oh no, not at all. So and then so I became an established group leader then at Würzburg University throughout the next years, and I started intense collaborations in Germany, which is a country where a lot of adhesion GPCR research is currently done. And so we started a scientific network that was quite important for my personal development as well, that is funded by the German Research Foundation, which is the main grant giver for fundamental research in Germany. And that brought me in close contact with people at the University of Leipzig, where I'm now a chair of general biochemistry at the medical faculty now. And somehow also paved the way of me being recruited there along the way. So I became a research professor, a Heisenberg professor in Würzburg still, and 6 months after that, I was recruited over to Leipzig to start heading a department of general biochemistry over here. So that's on the academic side or administrative academic side, the career side. And scientifically, that also meant that I got, well, half of it was that I built my own scientific network internationally and nationally, in which I could actually exchange ideas and have my ideas checked by others on the one hand side, and on the other hand, it's of course the science actually that we actually built up together, and that's been carrying, I would say, 2/3 of my lab currently, and I would say which is quite successful to work on adhesion GPCRs on very different levels. And that's where I'm now. So I'm now 7 years here since 7 years here in Leipzig.
I love it. Let's talk a little bit about the consortium or the organization that you are part of around adhesion GPCRs, because we've talked a little bit before we hit record about how important it is to know who are the key labs, who works on what in the GPCR field. And the adhesion GPCR field has its own universe. And in the
That's
true
true.
sense
Yeah.
of Dr. GPCR's mission is to really bring everybody together. So I'd like to know more about the Adhesion GPCR Consortium.
Yeah. So the Adhesion GPCR Consortium is a network of basically all labs that are actively working on adhesion GPCRs. And that was founded in 2011 out of an already existing tradition, hosted by a few labs that in the 2000s actually started to work on adhesion GPCR. So that whole field was born out of mainly of immunology research, because adhesion GPCRs are G-protein coupled receptors that have very extensive extracellular domains. So they were picked up back in the days by efforts to raise monoclonal antibodies against extracellular epitopes, basically. So a couple of these labs in the early 2000s that started to meet by annually, so every 2 years, was starting this very loose network. And I think I attended the second or the third workshop during my PhD time. So I got to know also the international players. And part of the charm of that network was that, so you had immunologists on the one hand side, and you had biochemists on the other hand, you had geneticists, and you had cell biologists. And the only thing about which they could talk was basically that central element, adhesion GPCRs, but they were actually illuminating and discussing this from very different perspectives, which was at times very complicated, because you had to accustom yourself with many different fields on the one hand side, but on the other hand, now looking back, I think this is also one of the one of the advantages of that field. And Mark, I didn't mention that there was any pharmacologist in the boat back in the day, because there weren't. And we had a couple of industry, not partners, but aficionados, I would call them, also invited and present at the meetings. And it was pretty clear that that field started out not as a venture of pharmacology, but of say cell biology or biology. And then, so over the years, when the discoveries actually came in and got some traction in the wider community, also more people, more labs joined, say, the idea of researching adhesion GPCRs, including pharmacologists. And at one point, we decided we need to have something, some institution basically that identifies us as the ones working on adhesion GPCR. So we founded this Adhesion GPCR Consortium, which hosts every 2 years these workshops still. Now, always in a 2-year cycle between Europe and North America. And that has been constantly growing ever since. So now we have about, I would say, 50 labs and about 150 persons that are registered as members, regular members or associate members of that network.
I love it. And I have to say, I visited the website and I think it's really nicely put together. It's so clearly you can tell that there is this group that's interested in adhesion GPCRs that comes together, and everything is so nicely laid out.
Thank you.
Including rules and regulations, which I thought was really phenomenal.
Thank you. Yeah. It's an interesting venture actually to be part of shaping a scientific field. And of course, it starts with the interest and the intrigue, and it also starts with exciting discoveries, but it also entails defining how you call parts of your receptors, how you call the receptors themselves, how you put them in relationship to each other, which I learned as part of, say, a scientific manager feature that I picked up along the way. Is very important too. Because it's the language that defines a field too. It's not only the people, it's also what you call the elements in that field.
You make an absolutely great point. We recorded a couple of podcast episodes regarding the history of G proteins and GPCRs. And it was so interesting. And we don't think about it. When I say we, I want to say the younger generation. Don't think about how is this protein named or how is this domain named. But essentially, when you think about it, somebody had to sit down, or a group of people had to discover this. Typically, multiple labs discover the same thing, and they have to sit down and decide what is this called.
Yeah.
And then it just takes on, which I think it's important, and we don't think about it in general.
Right, and it's very important that you don't have different names for the same thing, because that can, of course, lead to omission of discoveries or omission of papers. Let's put it very, very bluntly, right?
Yeah.
And also, basically, it's the loss of the of other researchers if they don't know about discoveries, just for the fact receptors are called differently. And so one of the major, I think, accomplishments of the Adhesion GPCR Consortium was that we negotiated with IUFA a general naming overhaul of adhesion GPCRs, and ever since then, all the general vendors of omics information now pull these names from the convention that we set up back then, and which renders you to immediately recognize, okay, so this project is about an adhesion GPCR because they all have the same prefix now. It's a little thing. It might seem boring, but in my view, it's pretty important.
It is. It is important. And, the one example that comes to mind is what I learned during this podcast series with Richard Permon, is that G proteins were not called G proteins in the beginning.
No.
So they had a completely different name, which escapes me. So if you go to PubMed, you can't go back and find those papers because nobody called them G proteins. And that might be a small detail when you're looking at a paper, but when you're looking at databases and you're exploring and you're using those databases for your own research and you're not picking up the right proteins, that completely makes it, difficult. And naming the protein, and agreeing on the name is very important.
Yeah, I agree. Yeah, and it also unites the community in the field, of course, too. So it was good actually to negotiate with very different interest groups within the consortium about this, because it's a very general topic. Everyone was convinced we have to agree on something. So it's also an exercise of whether you work as a community or not.
Yeah. And you do seem to work as a community.
I believe we work quite nicely. Yeah. So we're a nice consortium. We're a nice community. Yeah.
From what I've seen and what I've heard, I have to agree. All right. And the next meeting, and we have to stop for a second and talk about the next meeting, which
Sure.
is next year
Yeah.
in Mexico City, actually. If I remember
Yes.
correctly, October 23 to 24.
Yes.
Is
I
it
think so, yeah.
only reserved to people who are part of the consortium, or anyone is welcome to join?
Yeah, it's anyone is welcome. So this is one of the principles, and also say basically the freedoms that you still have if you are just an interest group and not an established, in part also commerce-driven enterprise like these big societal meetings nowadays. So we're still actually a very low threshold meeting, I would say, with very low registration fees. Everyone is welcome. The spirit I would compare at a meeting is similar to a Gordon conference. So there's a lot of unpublished research being discussed under Chatham House rules. And I think the reason for that is because we all have witnessed actually that because we have guests coming in, that at one occasion or the other, we learned something that we didn't know. Because again, we are not, many of us are not pharmacologists by training, yet we actually deal with the number one pharmacology target family in, say, at least human bioscience. So it's absolutely necessary that we get fresh opinions in, fresh blood in, that we don't establish borders to other neighboring areas of bioscience. And I think we have accomplished this over almost 20 years, I would say. And we'll try to maintain that spirit as long as we can. There's also some unwritten rule that a lot of junior researchers should present the research of their labs rather than the lab heads. And so we're trying to hedge our next generation. I think that has also worked pretty well in the past decade, I would say. Yeah, so everyone is welcome.
What a great spirit. I love it. Just as I love it. All right, let's get back a little bit to adhesion GPCRs. And I asked this from everyone on the podcast. What's your favorite GPCR?
So I
If you
probably
have
have to
one.
answer. Well, yeah, I probably have to answer. So yeah. So I'm divided. So in general, I'm interested in the entire family because they're so unusual. Every member is very unusual to the rest of the GPCR world. But the receptor subfamily that I've been working with most was called back in the day before we revised the nomenclature, laterophilins. And laterophilins are evolutionarily conserved adhesion GPCRs. Hence, I could actually research them, investigate them in C. elegans and Drosophila. And so if I would be pressed to name a favorite GPCR, would be the laterophilins. And there are 3 of them in humans and mammals, and 1 in a fly, 2 in C. elegans.
Wow, I love it. And any relationship between one in the fly and one of the 2 in C. elegans?
Yes, of course. I mean, structurally they're very tightly related. Even if you go all the way up the pedigree to the mammals, so they share most of the domain composition all along that 500 million year period, and they're all expressed in the nervous system and seem to fulfill certain functions there during development. So either embryonic development or also post embryonic development. And so we've been so my lab has been looking into these functions and the way how these functions are implemented on a molecular scale. Yeah, since now, almost 15 years now.
What are some of the methodologies that you use in the lab that you to study electrofilms or any other adhesion GPCRs?
Yeah, so over the years we've built up quite a big spectrum of technology. So it all starts, in our case, it all starts with in vivo experiments. So we do a lot of genetics and genomic engineering in the first place. So we take out the loci from C. elegans or from Drosophila, for example, generate null alleles. And so the first part of any project in my lab starts with trying to find what's wrong with the mutants, with the null mutants. So, and that actually opens basically a Pandora's box, because depending on where the receptor is expressed and what it causes, we need to learn new methods. So we started with electrophysiology because we'll have to interrogate neuronal messages, of course, neuronal excitability. So we did a lot of electrophysiology. We do a lot of imaging, starting from normal confocal imaging down to super resolution imaging using D-stone microscopy, for example. We do biochemistry in the last couple of years to identify ligands, interaction partners to these adhesive G-protein coupled receptors. We also started doing then in vitro experiments like coupling assays to better understand the downstream messenger pathways that are entertained by adhesion GPCRs, and so forth. So it's quite a range, and I would say we probably can cope with any regular or standard cell physiological or cell biological technique that's currently available, I would say. Yeah.
I love it. The other question I had for you, you mentioned in the beginning wearing all of these hats. How do you juggle all these responsibilities? Because you're chair of the department, you have your own group, you're also involved with the Adhesion GPCR Consortium. Again, I'm asking this from a perspective of our audience who may or may not be thinking about going into a career in academia.
Well, so there's the clever answer saying that I have a strict plan that I follow. So Mondays I do this, and Tuesday I do that. But that's not the earnest answer. I think I think I'm a good organizer, and I'm also well organized myself. So whenever demands come up in one of the say fields that you just mentioned, I try to make up time to tend to the problems, and also plan a little bit ahead strategically, but not much, but a little bit. So what is important for me and my lab in the next, say, 2 years or 3 years, and then I try to actually make time to talk about that. Second, I've had the chance and the joy to work with a couple of people that in my lab that are very good at organizing too, that actually take over a lot of the responsibilities and work on them independent from me. So I don't have to do everything myself, and I don't even have to organize everything from a say helicopter view perspective. So that's one of the boons of my scientific career, and has been, and hopefully will be also. And so somehow we make it work then along the way. So that's the earnest answer. Yeah.
No, and thank you for that. I think it's important. You make a great point about delegating, but also being as organized as possible, yet flexible.
Yeah.
And these three things are difficult, but you can learn to do them.
Yeah, I mean, if we've learned anything from the Corona period, then it's that, right? So don't make plans that go too far into the future, because you might be facing, completely different demands tomorrow, literally tomorrow, and then you have to make do with that, and somehow it works. Yeah.
Agreed. Have you ever considered going into industry instead of academia while you were finishing up your PhD?
Yeah, once. So when I was coming to an end with my PhD studies at Oxford, I at least applied for a conversation with a pharma company back then, and so I had that conversation, and after that conversation, I I was pretty sure that I should look for something else. So I don't even recall actually what the position was about. Some regulatory affairs topic, I guess. So something that I wasn't interested in, and so I made up my mind, and yeah, invested my energy on something else. I think you know, pharma companies can be a great workplace, depending on how your characters cut. Um, and if you're driven by curiosity about fundamental phenomena, it's pretty clear that you probably won't be happy in a pharma company that has very different demands, of course, on their own existence. And so it's just to find out what you're predisposed for, and follow this.
No, I agree. I think it's important to one, explore what's out there, to see what matches your personality, your needs, your interests. Otherwise, it can be a very long road of going to work in a position that you're not happy
Yeah,
and
painful.
doing things
Yeah.
that you're not, that you don't enjoy.
Yeah, I agree. Just only do what you're happy with, and what you like. I think that's where you're best at.
I agree. I agree.
Yeah.
All right. In the context of adhesion GPCRs, how has the view of pharma and biotech changed over the years? We've established that they're different and very complicated targets, considering that they can be anywhere between 600 to 6,000 amino acids in these receptors. But from your perspective, how much has the field advanced? And do you know, have you seen any changes in the interest from pharma or the private sector into working on
Yeah.
adhesion GPCRs?
So starting with the scientific side of things, yes, there was a big change in how adhesion GPCRs are perceived nowadays by the entire community, including pharma companies, and that was brought about by basic research. So none of that, I would say, was contributed by pharma companies, but it's harvested by them. So it was purely academic labs that have discovered first that adhesion GPCRs can couple to G protein pathways or other, say, intracellular outlets, which wasn't clear at the time when I joined the field. So there was a big skepticism of whether they are just adhesion molecules and use the 7-transmembrane domain as a very firm anchor of the adhesive properties to the cell membrane. So I think people can look this up as working hypotheses in the mid-2000s. And so showing that adhesion GPCRs truly can engage in G proteins is a big paradigm shift. Secondly, that was brought about by discoveries of how their signal transduction can be instigated, and that they carry something that is known as a tethered agonist, or a Stachel, German word for stinger, that was discovered by 2 labs here in Leipzig about 10 years ago, which for the first time made tools available to stimulate adhesion GPCRs at will. So because one of the problems was that there was an agonist known for an adhesion GPCR, and so how can you actually start pharmacological assays if you don't have an agonistic versus a non-agonistic situation, right? So that was a second very important discovery. And third, I would say the fact that adhesion GPCRs play roles generally in developmental processes. It could be embryonic development, as I said, but it could be something very specifically post embryonically, like the formation of a synapse, or the formation of alveolus in the lung, for example, or how the skin is structured. In all of these processes, adhesion GPCRs seem to play a pivotal role, and hence the breakdown of these signals may contribute to a lot of diseases, human diseases, that may be at least associated, if not causally linked, to adhesion GPCR signaling. And I think this is the third thing that people now started to realize. So we have a targetable GPCR class that is involved in a whole lot of other processes than a regular, say, the class A GPCRs were found. And I think that, of course, has changed the perspective on that family of GPCRs quite a lot. So there's a lot of interest. That was the second part of the question. There's a lot of interest from industry in adhesion GPCRs, perhaps little bit for different reasons. So one reason is that adhesion GPCRs, because of their usually long extracellular surfaces, make for excellent epitopes for any disease. And now with the advent of immunotherapeutics in cancer therapy, for example, like CAR cells, CAR T, and NK cells, or cancer vaccinations, basically has uplifted the status of adhesion GPCRs as to the point that they are now, I think, actively researched for basically targets for these drugs, not necessarily for their GPCR signal transduction route, but for basically their reservoir of epitopes. And secondly, on the other hand, people, of course, are trying to now harness or hijack the signaling routes, the metabotropic signaling routes of adhesion GPCRs too. So first screens have been initiated based on the now available downstream pathways that can be assayed now. So I think I think we're still in the linear phase of interest from pharma companies, but I expect it to spike in the near future.
Agreed. And I also, I'm not an expert on adhesion GPCRs, but I know many scientists working on adhesion GPCRs who came and presented at all of our different Dr. GPCR events. And every time someone came over a period of time, you could see that there are new tools, and our understanding of how these receptors function is growing. And as I mentioned, we're developing more and more tools to study these. And also, I mean, electrophysiology is beautiful, but you can't run that many samples that you can run in a plate.
No, you need some systematic, you need tools to systematically go through a single homolog, basically with all its nooks and crannies, but also through the entire family, which is quite large, admittedly. So it's 32 human homologs, and they are very vividly spliced. So we probably deal with hundreds, if not thousands, of isoforms for these 32 human adhesion GPCRs. Yeah. So you need some systematic assayability to go through them. Yeah, I agree.
In your opinion, what are some of the tools that we need, and following up to what we were just talking about, that we have a little bit more understanding as to how these receptors function? We're developing tools to assay them in a high throughput-ish fashion.
Yeah.
What else is missing in your opinion?
So I think one of the reasons why for many decades adhesion GPCRs weren't used by academic pharmacologists and also pharmacology as a commercial venture is that there are some irregularities on how these GPCRs actually work, how their signal transduction is actually stimulated in the first place. And this is very unusual. So as I said, so as opposed to say class A or class C receptors, or even class B1 receptors, which are activated by soluble compounds, adhesion GPCRs seem to be activated by non-transient, so persistent adhesive interactions. So you can't just simply wash some soluble agonist over them, with the exception of these tethered agonists that you can synthetically make nowadays, but it's still a very artificial situation of doing this. So one of the main bottlenecks is how to construct a screening situation, a screening paradigm in which you have a natural adhesive interaction engaged, and you can read out what happens, or what is instigated by that adhesive interaction. So this is a formidable intellectual question. So how you do this in a high throughput format. And that this is not solved yet. So this is something I think that our field has to concentrate on. So that's the first thing. The second thing is the concept that adhesion GPCRs are adhesion recognizers is fed by the fact or fed by the discovery that they seem to function as mechanosensors. So I think my lab was the first one that showed in Drosophila, together with the lab of Robert Kittel back then in Würzburg, that laterophilins, so the adhesion GPCR type we are interested in, is physiologically necessary to recognize mechanical stimuli. And this was actually promptly followed up by a pharmacological demonstration of this fact. So they are very unusual in terms of what the ligands are, and second, of what type of stimulation, physical stimulation, they recognize. And I think people have to take this into account when setting up screening platforms for them. And that's probably also the reason why pharma industry lost interest, because in all their screens in the 90s, 80s, 90s, 2000s, they came out empty-handed, because you can't really actually activate adhesion GPCR activity by washing something over them.
Yeah, no, I agree.
Big,
I agree.
big challenge, yeah.
Big challenge. I was earlier this year, I was at an Integrin Gordon conference, and I was very much delighted to see all the effort put into mechanosensing in the context of integrin and integrin signaling. So I wonder if the adhesion GPCR field can learn from a methodology perspective from the integrin field there.
Most definitely, yeah. I mean, there's a couple of molecules that have been proposed to act as mechanosensors, very similar to adhesion GPCRs, and I think we are not the first field to place our proteins of interest or receptors of interest in that context, and I think there's a lot to learn from the knowledge that other fields have already actually obtained on that.
Yeah, wonderful.
One of the other, maybe one other thing that I would like to point out is, and we've actually discovered this in a project that was just published by my lab and the lab of Nicole Scholz here from Leipzig this year, that adhesion GPCRs may not only be receptors for cell autonomous signals, so not for signals that are perceived by the same cell that produces the adhesion GPCR, but also for cell non-autonomous signals. So for signals that occur in other cells than the one in which the adhesion GPCR is synthesized and expressed. And because what we found is that adhesion GPCRs can actually shed part of their extracellular regions by some biochemical mechanisms that are at play in most of adhesion GPCRs. And these shed N-terminal fragments can then act as ligands for other receptor systems, for example, for toll-like receptors. So when we think about adhesion GPCRs, we should not only think about them as GPCRs, but they may also be a source for ligands for a couple of other systems, receptor systems that are not even related to G protein coupled receptors. So they can be ligands too.
I love it. I have goosebumps. I love it. This is really, there's so much to do. This is really great. All right. Top 3 aha moments that you had as a scientist that shaped your trajectory.
Yeah. I think so. Looking back, so aha moment implies that I realize at the moment when something happens that this is the moment, which I would say I rarely had. I couldn't actually probably put my finger on one, on either one of them. But looking back, I think one of the aha moments, for at least for my personal career, was that don't follow the beaten track because it might not exist or it's too crowded. So just follow your instinct, do what you like, as I said before, because that's what you're best at. And that has worked out for me so far. So that was probably the first one. The second one with respect to pharmacology is that, at least for the field of adhesion GPCRs, that pharmacology is actually worthless in the absence of physiology. So you first have to somehow understand in which context a receptor works, and then actually it makes sense to, and it's even necessary to get in pharmacological expertise, but not the other way around, because I think that would have obscured a lot of the discoveries made for adhesion GPCRs in the past. And yeah, follow your interest, don't follow fashion. Just be just do what you're really passionate about. I think because it's a long career. So if you really want to follow through with a career with 40 years, that's a long while, and you should actually try to maintain your interest in that. And you can't do that if you just follow a recipe given to you, handed to you by somebody else. It's probably not very original, but these were the three things that I would stress.
I love it. Any other advice to junior scientists who want to contribute to the field?
Well, get in touch with somebody who's already on the project, just to basically get informed, not to negotiate any boundaries, because in the adhesion GPCR field, this is not necessary, and it's also not suggested. But just so you know who your colleagues could be, so you can run your ideas by, so you get some critical input, which is very important. I think that's quite important, and turn up at the workshop and introduce and present the stuff, because there's going to be a lot of people who can help you, and you can help too, most likely.
That's great. Well, last question: if and when you have job positions open in your team, where can people find you other than on the Dr. GPCR ecosystem?
Right. So so we have a mailing list of the Adhesion GPCR consortium. There's a website as well, adhesiongpcr.org, one word, .org, where we advertise positions. And on my lab page, you can find this as well, which I think is probably linked to this interview then later on.
It will be. It will be. With this, Tobi, thank you for your time. Don't go anywhere. Let's let people wonder what we talk about after we stop recording.
Okay. Thank you very much.
It was a great pleasure having you.
Likewise. Thank you very much.
Can I ask you for a favor? Please subscribe to our YouTube channel. Many of you come back and watch our videos, but aren't subscribed. Having more subscribers will help us get you more content. Also, mark your calendars and join us in person for the inaugural Endocrine Metabolic GPCRs meeting taking place in Birmingham in the UK between April 22nd and 23rd. Thank you for joining us and listening to this Dr. GPCR podcast episode. We would like to thank our guests, our team members, Attila, Ines, Montserrat, Ivana, Andreina, and Balin. If you like our podcast, leave us a 5-star review on Apple Podcast. You can also leave us a testimonial at drgpcr.com/testimonials and subscribe to our monthly newsletter today. Another way to support us is to share your favorite Dr. GPCR program with your colleagues and network. With any questions or suggestions, email us at hello@drgpcr.com. And until next time, stay safe.